Everyone's heard of COVID-19, a respiratory infection that's affected more than 200 million people globally. One of the most common symptoms of COVID-19 is anosmia, or the loss of smell. Anosmia has affected more than 53% of COVID-positive patients and is one of the first few symptoms seen in COVID. But how do we lose our sense of smell? And why do some people lose their sense of smell, but not others? To answer these questions, we must first understand what viruses are. Viruses are extremely simple. They enter a cell, make copies of themselves, kill the cell, and then go on to infect other cells. Since they live and reproduce inside the host cell, they're known as intracellular parasites, much like a robber hijacking a house. Most viruses have a genome and a protein code, but some viruses, like the SARS-CoV-2 virus, also have an outer layer called an envelope. Viruses enter the host cell using spike proteins on their envelope, and these spike proteins bind to specific receptors on the host cell, allowing the virus to get in. The type of spike protein a virus has determines the type of host cell it affects. So how is all of this related to the loss of smell? Let's first look at how we smell in the first place. Olfaction occurs at the roof of the nasal cavity where the olfactory epithelium, or the retina of the nose, is located. The epithelium consists of three types of cells: support cells, basal cells, and olfactory sensory neurons. Basal cells are nasal stem cells that give rise to olfactory sensory neurons. The support cells, including sustentacular cells, give metabolic and structural support to the neurons. The neurons have many tiny cilia at their ends that contain receptors. These receptors bind to odor molecules, allowing for odor detection. Once odor is detected, an action potential is generated in the sensory neurons. The signal is sent to the olfactory bulb, which relays it to the brain for odor perception or olfaction. Now that we have understood how we smell, let's delve into how the virus causes anosmia. The spike protein found on the virus interacts specifically with ACE2 receptors and TMP or SS2 proteases on the host cell. These interactions cause a series of conformational changes that result in viral fusion and entry. Unsurprisingly, ACE2 and TMP or SS2 are highly expressed in the olfactory epithelium, specifically in the sustentacular cells. Damage to these support cells indirectly damages olfactory sensory neurons, leading to a loss of smell. This also results in a major loss of cilia, which are necessary for odor detection. But the immune system also plays a role here. Viral infection is often accompanied by a rapid immune response, leading to inflammation and destruction of the olfactory epithelium. Specifically, IL-6, a pro-inflammatory cytokine, activates apoptic pathways in the olfactory epithelium, leading to the inhibition of smell. The scar tissue formed after inflammation can also hinder future regeneration of the olfactory epithelium. Finally, the virus can also damage the olfactory bulb and nasal stem cells, which are both critical for olfaction. Most patients recover in 10 days, but this can go as high as 70 days. A quick recovery rate is found in patients where the virus affected non-neuronal cells in the olfactory epithelium, whereas prolonged recovery is associated with damage to the olfactory sensory neurons, nasal stem cells, olfactory bulb, or the cilia. But why are some people more likely to get anosmia compared to others? Females and the elderly, for example, are most susceptible to COVID-19 anosmia. This relates to the severity of infection, number of ACE2 receptors, and regenerative capacity of the olfactory epithelium. These factors depend on age, sex, disease, and degree of inflammation. Patients with mild infection reported higher levels of olfactory dysfunction compared to those with severe infection. In fact, 47.7% of patients with mild to moderate COVID-19 reported olfactory dysfunction. The mechanism is not entirely known, but could be due to the difference in the type of infection seen in severe and mild patients. There are two types of COVID-19 infection: upper and lower respiratory. Upper respiratory infection, including the loss of smell, is more commonly seen in milder cases of infection. Not surprisingly, females are more likely to have milder cases of infection because estrogen downregulates ACE2 expression. Milder cases of infection are associated with upper respiratory infection, including the loss of smell, and this is why females have higher prevalence of COVID-19 anosmia than males. Last but not least, older COVID-19 patients are less likely to report anosmia compared to younger patients. This is because older individuals have a thinner olfactory epithelium due to its degradation by age. This results in fewer sustentacular cells, fewer ACE2 receptors, less viral infection, and thus less prevalence of olfactory dysfunction in older patients. Now we know that losing your sense of smell isn't really something to worry about, apart from not being able to smell your favorite food or perfume. This loss of smell is primarily caused by damage to the olfactory support cells, and the degree to which the epithelium is damaged determines severity of infection, which in turn determines who has a loss of smell and for how long.